Hi guys, welcome back to Screencastifying with Miss MacGyver. Today we are talking about 17.2, so transcription being the direct link between DNA and RNA, and eventually how we get to amino acids and proteins. So transcription is the first stage of physical gene expression. So we already know that DNA composes our genotype, and proteins or polypeptides is the phenotype. So, how do we get there from DNA all the way out to physically seeing the traits that people have? And we do that by first starting with transcription. The molecular components of transcription. So, RNA synthesis is catalyzed by RNA polymerase. It acts very similarly to DNA polymerase. It actually pries the DNA strands apart and hooks together the RNA molecule. So it's kind of doing what replication is doing, but it's RNA polymerase, so it's making RNA and not DNA. The RNA is a complementary strand to the DNA template, so it uses the same base pairing rules, except for the fact that uracil substitutes for thiamine. So A and U pair together and not A and T in RNA, because RNA does not have a thiamine, it has a uracil. DNA sequences are where the RNA polymerase attaches to is called the promoter. In bacteria, the sequence signals the end of transcription is called the terminator. The stretch of DNA that is transcribed is called the transcription unit. So in bacteria, we have our promoter and our terminator all together. In more of a eukaryotic cell, the promoter attaches to the promotion sequence, goes down, so it starts to separate out. It starts moving down the DNA, creating an RNA transcript in the process, and then resealing the DNA back together at termination. The synthesis of RNA transcript happens in 3 different stages. So we have initiation, elongation, and termination. RNA polymerase is the binding and initiating system of the transcription process. Promoters on the DNA signal the transcription start point and usually extends several dozen nucleotide pairs moving upstream from the start point. Transcription factors mediate the binding of RNA polymerase and the initiation of transcription. The completed assembly of transcription factors in RNA polymerase II bound to the promoter is called the transcription initiation complex. This is also signified by something called the TATA box. It's a series of TATA in the DNA strand that signal that it is the promoting region for that specific gene. It is crucial in forming the initiation complex in eukaryotic cells. Without that TATA or the TATA box, we cannot move forward with transcription. So here you have that TATA sequence, which is the start of your promoter, which then would bind in that RNA polymerase into the promoting region and then start the RNA transcript. The elongation of the RNA strand. So RNA polymerase moves along the DNA and it untwists the double helix at about a rate of 10 to 20 bases at a time. Transcription progresses at a rate of 40 nucleotides per second in eukaryotes. A gene can be transcribed simultaneously by several different RNA polymerases. Nucleotides get added to the 3' end of the growing RNA molecule. So we're still paying attention to that 5', 3' end of DNA. This is what it would kind of look like in large. So we unzip a portion of that DNA molecule. The RNA starts binding, so you can see A and T we still need to get that A in RNA, so T will still match with A, but in RNA, we can't carry a T, we carry a U for uracil. So A and U pair together. The termination of transcription is a series of mechanisms that is different in single-celled organisms and in multi-celled organisms. In bacteria, the RNA polymerase stops transcription at the end of the terminator, and the messenger RNA can be then translated without any further modification. Because remember, in bacterial cells or prokaryotes, there is no differentiation of region. There's no real membrane-bound organelles, so the DNA is in the cytoplasm. But in eukaryotic cells, the RNA polymerase II that just finished transcribing that sequence has to then pass it along to a transcript that gets released at about 10 to 35 nucleotides past that end sequence. So it then can go through a priming stage so it can leave the nucleus. After transcription, the enzymes inside the nucleus modify the pre-messenger RNA through a step called RNA processing before the genetic message can then be passed out to the cytoplasm. During RNA processing, both ends of the primary transcript are usually altered or removed. And some interior parts of the molecule get cut out and then others get spliced together. So genes are very, very long, and one gene can consist of several coding and non-coding regions. We don't want those non-coding regions because they don't mean anything. So this is the processing step that cuts those out and then re-puts the RNA back together. The alteration of the messenger RNA ends. Each end of the pre-mRNA molecule gets modified in a particular way. The 5' end receives a cap, while the 3' end gets a poly-A tail. These modifications share several functions. First, they seem to facilitate the export of RNA out into the cytoplasm. So we're still inside the nucleus at this point. The second is that they protect the messenger RNA from enzymes that want to break it down. And the third is to help the ribosomes attach to the 5' end during translation. So this is what it would look like. You have your 5' cap and then your poly-A tail. Split genes and RNA splicing. So some eukaryotic genes in their RNA transcripts have long non-coding stretches, like I said before, of nucleotides that lie between the regions that actually code for that polypeptide. These non-coding regions are called intervening sequences or introns. The other regions are called extrons because they eventually will become expressed and translate into that amino acid sequence. RNA splicing removes the introns and then joins the extrons together that creates a messenger RNA molecule with a continuous coding sequence. We want to get rid of that mumbo jumbo that we don't need. So we remove the introns and then splice together the extrons. So here, the light color is your intron. We don't want that, so we cut it out and then reseal together those extrons. In some cases, RNA splicing is carried out by a spliceosome. It consists of a variety of proteins and several small nuclear or SNRNPs that recognize the splicing sites. So looking at it kind of visually, this is our spliceosome. It catches one end and the other, cuts it, and then at the very end, we splice together those two extrons. Ribozymes are those that catalyze RNA molecules that function as enzymes to splice RNA. The discovery of the ribozyme rendered the belief that all biological catalysts were proteins. There are 3 properties of RNA that enable it to function as an enzyme. First, It can form a three-dimensional structure because of its ability to pair with itself. Second, some bases of RNA contain functional groups that may participate as a catalyst. And three, RNA may hydrogen bond with other nucleic acid molecules to protect itself. There is function and evolutionary importance of the introns. So you're probably sitting here listening to this like, well, if we're getting rid of the introns, why do we even have them? Well, some introns contain sequences that may regulate the expression of certain genes. Some genes can encode for more than one type of polypeptide depending on which segments are treated as extrons during the splicing process. This is called alternative RNA splicing. Consequently, the number of different proteins an organism can produce is actually much greater than the number of genes that we have. So for one particular gene, the intron may not be important, but that same intron can be an extron for another gene. Proteins often have a molecular architecture to them that consist of discrete regions that are called domains. In many cases, the different extrons code for different domains in a protein. Extron shuffling may result in the evolution of a new protein to be produced. So looking at it this way, here are our extrons, our introns, extron, intron, extron. Transcription happens, and then we can shuffle these around that may result in a different protein actually being built. All right, we are going to stop here. Your worksheet that is due Friday by midnight does have translation on it, but you're going to get 17.4 on Friday. All about translation, so you guys will be fine. You guys will be able to go through and do the transcription part without any major issues. So for now, I'm going to say adios. And if you guys have any questions, as always, email me.